Hi everyone, and welcome to our demonstration of ELISA disease detection modeling for SARS-CoV-2, the virus that causes COVID-19. In this video, we'll show how two types of ELISA tests, antigen detection and antibody detection, are used to diagnose infection and measure immune response. ELISA stands for Enzyme-Linked Immunosorbent Assay. ELISA is a laboratory technique that uses antibodies to detect specific proteins, known as antigens, in a patient sample. Because every antibody binds to only one unique antigen, ELISA tests are highly specific and widely used to detect diseases. These diseases include HIV, influenza, Zika, Lyme disease, and now SARS-CoV-2. When a person becomes infected with a virus, viral antigens appear first and trigger the immune system to make antibodies. Over time, as antibodies increase, they help destroy the virus and the antigen level falls. That means an antigen detection ELISA is best for finding an active infection, while an antibody detection ELISA tells us if someone has mounted an immune response or has had a past infection. Our group will thoroughly demonstrate both. We'll show how antigens and antibodies interact in each model, describe what the color changes mean, and answer some key questions. Let's go ahead and start with the antigen detection ELISA, showing how the test detects viral particles in a patient sample.
The goal of antigen detecting ELISA is to determine if viral proteins are present in a patient sample, in this case, SARS-CoV-2 antigens. For step 1, we add a patient sample to the well. This sample may contain viral proteins, but also many other proteins. All of them stick or absorb to the plastic surface of the well. We rinse with a wash buffer to remove unbound materials and block any leftover open areas. For step 2, we add the primary antibody. This antibody is designed specifically to bind SARS-CoV-2 antigens. For example, antibodies against the virus's spike protein. Only matching antigens will bind tightly. Everything else is washed away. This step shows the concept of antigen-antibody specificity. For step 3, we add enzyme-linked secondary antibody. This antibody recognizes the constant region of the primary antibody and is linked to an enzyme such as horseradish peroxidase. It binds wherever the primary antibody is attached. After washing again, only bound secondary antibodies remain. For step 4, we add the substrate. If the enzyme is present, it catalyzes a reaction that turns the substrate blue, producing a visible signal. A blue color means the viral antigen was in the sample, confirming an active SARS-CoV-2 infection. If the well stays clear, no antigen was detected. Now that we've seen how antigen detecting ELISA finds the virus itself, let's move on to how an antibody detection ELISA measures a person's immune response.
The antibody detection ELISA uses the opposite strategy. Instead of looking for the virus itself, it's looking for antibodies produced by the immune system after an infection or during an ongoing infection. Step 1 is to coat the well with purified viral antigen. Here we're using the SARS-CoV-2 spike glycoprotein that was produced in a lab. The antigen binds to the well surface and a wash step removes any unbound material. Step 2 is to add the patient serum. If the person has been infected, their serum will contain antibodies that recognize and bind to the spike protein. Antibodies from uninfected individuals won't bind and will be removed during the wash step. Step 3 is adding the enzyme-linked secondary antibody. For human samples, this is often a goat anti-human antibody attached to the same type of enzyme that was used before. It binds to any human antibodies that are attached to the spike protein. Another wash step here removes the excess. Step 4 is to add the substrate. If anti-SARS-CoV-2 antibodies are present, the enzyme reacts with the substrate and turns the solution blue. The color change confirms that antibodies are in the patient's serum, meaning they were infected previously or are currently undergoing an immune response. However, a positive color doesn't necessarily mean that there is an ongoing infection because antibodies can persist long after recovery. Next, we're going to discuss how to interpret results correctly and the importance of controls in both tests.
To ensure the validity of each ELISA test, we include both positive and negative controls. The positive control already contains a known antigen or antibody. This should always turn blue to show that our reagents and enzyme system are working correctly. The negative control remains clear, proving that no contamination or false binding occurred. The patient wells are then compared to these controls. Patient A matches the positive control, meaning a positive result. Patient B is clear, meaning negative. Here's how we interpret those outcomes. When both controls behave as they expect, the test is trustworthy. If a patient's sample turns blue like the positive control, it means the target, either the antigen or the antibody, was present. If it remains clear like the negative control, it means there was no detection of any antigen or antibody. Wash steps are one of the most critical parts of ELISA. Each wash removes any of the unbound antibodies, enzymes, or substrates to prevent the false positives. This ensures that only specific antigen-antibody interactions cause the color change. This infection timeline shows how the body's immune response changes over time. The red curve shows the viral antigens. They appear first and fade once the infection clears. The blue curve shows the antibodies. These rise later and can stay high long after recovery. That's why an antigen ELISA cannot diagnose infection on day 35. By then, the antigens are gone, but the antibodies remain. So now that we understand when antigen and antibody appear in the body, let's look more closely at how ELISA actually detects them. Both the antigen and the antibody tests rely on something called a secondary antibody, which makes the reaction visible. But have you ever wondered how the same secondary antibody can recognize many different primary antibodies? Let's take a closer look. Here are two different rabbit primary antibodies. Even though each one binds a different antigen, either red or purple, their constant regions, the stem parts of the Y, are identical because they're both rabbit antibodies. The goat anti-rabbit secondary antibody recognizes that constant region. It doesn't care what antigen the top part is binding. It simply attaches to the constant region that all rabbit antibodies share. That's why one secondary antibody can detect many different rabbit primary antibodies. The orange dots represent the enzyme linked to the secondary antibody, and when substrate is added later, that enzyme is the reason for the color change to signify a positive result. A blue color in an antibody ELISA means that antibodies are present, but not necessarily that the infection is active. Antibodies can persist in the bloodstream for weeks or months after the recovery. To summarize, the antigen ELISA detects the virus itself, showing an active infection. The antibody ELISA detects the body's immune response, showing current or past exposure. Together, these two tests help scientists confirm infection, monitor recovery, and measure immunity. Thank you for watching our ELISA disease detection demonstration.